I think it's great. I got her to actually, I've got a clicker. Oh, let's try it out. I got you to quote Spider-Man, and that's pretty cool, right? If you can't get anyone to quote Spider-Man. So there we are. Take a picture of everyone. And put it on Twitter, because that's what we do. Passes the time between projects. Okay. So I'm going to talk to you, I haven't changed the title of this slide, so I apologize for that. I got up at 3 a.m. this morning in order to come out here, and I probably was half asleep on the plane. But in essence, I'm going to be talking about a number of different projects that we're currently involved in, and hopefully give you some examples of how the work that we're doing has been empowered by next-generation sequencing, but also how we're kind of trying to reinvent the paradigm of what's capable and possible within this field for looking at environmental consequences of the microbiome. So I work on a number of different aspects. One is the home microbiome project and the hospital microbiome project, which are two new initiatives that we're pioneering, and I'll give you some results from those today. And then I kickstarted the Earth Microbiome Project around 3 years ago. I'm going to talk a little bit about what we've done there, how we've used those results to transform certain aspects of modeling microbial dynamics across the planet. And then when Rob Knight comes in this afternoon, he's going to give you a little bit more detail on the statistics and numbers that we've been generating in that project, which are, I still think, quite impressive. So, as we all know, hopefully, we are a microbial life form. We're almost a fomite, just a lump of flesh that came along in the last 5 million years and has been heavily colonized by microbes, which have, basically taken up root in every facet of our body. 2 to 6 pounds of your body mass is made up of bacteria and protists and archaea. And that 2 to 6 pounds is probably one of your most vital organs. Without it, you suffer from severe, if you didn't take any vitamins, you'd suffer from severe nutrients loss. You'd also suffer from a much heightened increased probability of getting an infection. You can become very sick and possibly die. So they're vital to our everyday life, even with supplements. And they colonize every aspect of our body, and it's very important that we understand those. But more importantly, they've evolved with us. They've evolved with us over the last 5 million years to the point where we absolutely rely on these organisms for virtually every facet of our day-to-day existence. From digesting our food to maybe even making decisions for us mentally, to changing the way we interact with different people. And some of the evidence I'm going to show today points to those kinds of sources of the microbial influence upon our lives, both social, physical, and medical. Now, one of the facets of this is, and this is something I've been getting into recently, so I like to introduce it in this, but is the influence of microbiology on your thought processes, on your neurological behavior. Now, I'm a father of an autistic child, and I understand the impact of potentially microbiological results and implications on his behavior and on his lifestyle. And that, to me, has fundamentally changed the way I have seen in terms of the way bacteria influence behavior, the way bacteria influence disease states in the body, and the importance of really pushing forward and understanding the complexities of those interactions. And this comes around from what we call the gut-brain axis, the fact that there are 2 to 3 pounds of bacteria in your body, in your intestine alone, has to have some kind of implementation. Those guys are generating huge numbers of metabolites, huge numbers of chemicals that get into our intestinal cavity and then can transcend the gut-blood barrier, move into our bloodstream, and influence certain aspects of our neurological behavior. Even without that, if we sever the vagus nerve between the gastrointestinal system and the brain, we start to reduce the likelihood that the microbial contents of the intestine can influence behavior. And you see this if you put mice in a situation where they should be nervous because they're up very high and they could fall left or right of a very narrow beam. They get very anxiety-ridden. Now, if we remove the bacteria from their gut, they lose that anxiety. Also, if we cut the vagus nerve between the gut and the brain, they lose that anxiety. So the microbes are automatically teaching these bugs, these mice, that if you walk across a very narrow area and you fall, that's a bad thing. So they're actually influencing the behavioral responses of these animals. And we have an enormous amount of evidence now that it transcends into human beings as well. After all, just a larger mammal. And so this is driving forward our research objectives. In autistic children, the influence of a pathogen or a microbe can have a significant effect upon their behavior. This girl's drawn a lovely picture beforehand. She's about 7 years old, and she's very good with her pencils. Now, if she gets a streptococcal infection, like a strep throat infection in her respiratory system, then she undergoes a severe neurological relapse to the point where she no longer has control of her fine motor skills. She loses her ability to conceptualize patterns and ideas, and that has a significant impact upon, obviously, her social status and her life. If we give her a healthy, or what I would consider an unhealthy, dose of antibiotics, we can restore her neurological behavior to what it was before. So she's now able to reposition herself and reduce that neurological deficit. So we know that if we affect the microbiome, we can affect behavior. We can also affect neurological issues. Work we've recently been collaborating with Sarkis Mazmanian out at Caltech, and he and Paul Patterson have been showing some fantastic results on the reason certain children may be more susceptible to these behavioral modifications, such as gut permeability caused by immunological and stress responses during pregnancy. So the child now has a semi-permeable gut membrane in which more of the metabolites which the bacteria are producing can get into the bloodstream. This leads us to the conceptualization of dysbiosis, how the bacteria in your body may have a reduced symbiotic relationship with you. And if that's the case, we need to track down where that's coming from. Is that coming from the way we live nowadays? We live in an indoor environment where we're constantly exposed to each other's microbiomes, rather than an outdoor environment where we're sharing the microbiome of the animals and plants and soil that live around us. Maybe that's influencing the sudden increase in neurological deficit disorders that are occurring and have been accelerating over the last 20 to 30 years. So we're very interested in trying to explore and push that. And we've created a national consortium to try and push forward for clinical trial practices and are moving into trying to utilize next generation sequencing in order to give us the hypothetical baseline against which we can now generate that information and create a knowledge base which we can utilize to move forward with some real clinical strategies. Now, this also comes to the changing face of medical treatment. I mean, if you went into a surgeon 200 years ago, you'd probably not really come out alive, or you might come out very, very sick or with a severe infection. Most women didn't survive childbirth even 150 years ago. It was an incredibly dangerous time. I'd say most women, but there are obviously a lot more people on the earth alive today, so there must have been some survivors. But we moved on from this conceptualization of surgery being a horrible and very painful instance to where we now have a clean medical practice. Over the last 100 years, we've learned to deal with a sterile cultural environment where we sterilize the hospital room, we try as much as we possibly can to eradicate the bacteria, eradicate the viruses and the protists that may infect a patient during surgery, and we've had wonderful results. We've managed to save hundreds of millions of lives by using this approach. But this is not the only story. We can't just continually move forward when we're still losing 100,000. This is just Americans, and I'm English, so I care about you people. But we're still losing 100,000 Americans a year to infections that you get during surgery. Okay? So the sterile procedures that we perform, no matter how good you think they are, they're not quite good enough. We need to explore other opportunities, other avenues in parallel. Obviously, there's a huge barrier to this. If you invoke a new strategy and somebody dies, there's an immediate cessation of activity on that strategy. You cannot move forward very quickly. You have to be very careful about how you push this forward. You have to have very good animal models. You have to move forward very carefully with treatment. You cannot rush into these. But we also can't be complacent. We need to move forward with a strategy which enables us to develop those clinical strategies appropriately. Florence Nightingale had this down 200 years ago. During the Crimean War, she noticed that when she went around hospital buildings, if she changed the way the hospital strategy was working, such as by even just by opening windows or increasing the amount of light or moderating the temperature of the room, as she called it, she could see a significant improvement in the mortality reduction in those wards. So she was already seeing that there was a bigger picture. It wasn't just about keeping this patient clean and isolated. You also had to do other things like fresh air. What's wrong with fresh air? Fresh air is a great thing. Open a window, increase the microbiological diversity. And Jessica Green at University of Oregon has really been pushing this idea. She's shown that when you open windows in an environment, you reduce the number of potential pathogen-like organisms in the air. You increase the microbiological diversity of that air, and that can't be bad. It seems to have a positive benefit and beneficial effect. So we need to move away from just killing everything off. This brings me on to the home microbiome project. In this study, we were pushing to try and understand what happened when you moved into a new house. When you, with your microbiome, moved into a house with its microbiome. What happened to you and the house? So did you start looking like the house? So did you absorb all those house-borne microbes and become a different person? Or did the house start looking like you? Did you stay mutually exclusive, or did you reach some kind of new state where you were both fundamentally changed by the collision of those microbes? And we worked with citizen scientists in this project, and you'll hear a little bit more about this if anyone's coming along to ASM on Sunday, where the postdoc Daniel Smith, who was fundamentally involved in this work, is going to be presenting in Jonathan Eisen's Citizen Science Project program, because this is a fantastic opportunity for us to reach out to the public as well and try and educate people on the importance of microbes and how we can now interrogate them with our new techniques. So we sent them little kits and little freezers, enough kits so they could swab themselves and any other mammals, including their children, in the house daily for a 6-week period. And they did that on 3 locations on each human or animal, and then also on certain high-touch surfaces in those environments. And we monitored the temperature, the humidity of those environments. So, here in the bathroom, you can see spikes in relative humidity as this house is having a shower every day, and that changes the environment in which those microbes find themselves. And we see significant relationships with that. Here you have peaks in humidity in the bedroom. I should skip over that. And we're trying to understand those relationships, what that means for the microbiome, how it could mediate and push ahead and try and help us to understand how the bacteria colonize those environments. Now, this looks like a big splodge or something your cat would throw up, but essentially every dot in here is a sample. And in those samples, there can be hundreds of thousands of bacterial species, taxonomic OTUs. This is done with 60-nose ribosomal RNA V4 sequencing using the MiSeq platform. And this is done using the Earth Microbiome Project standard protocols, which you can find on earthmicrobiomeproject.org. And all the dots out here on the edge are these little lines that they're going to. Click one forward. Those are OTUs. That's a bacterial species. And the lines that come away from it, those are lines that demonstrate where that bacterial taxon was found in the environment. So if you see here, the yellow dots are samples from home surfaces, and the green dots are from people, and the red dots are from animals. And what we were doing here was trying to understand that interrelationship. And what we found out is, if you have a home with animals, and these are colored by the home that they came from. If you have a home with animals in them, you have an incredibly increased diversity and also a massive conduit by which the microbes can pass through. Now, my postdoc doesn't work quite as fast as Rob Knight's postdocs, and Rob's Knight postdoc just published a paper on this in eLife, where he looked at families with dogs, not across time, but across individual households, and did a lot of this investigation. And in his study, they found that if you had a dog in the home, it increased the microbial transfer rate between you and your spouse. So you actually shared more microbes with your spouse. But interestingly, if you had a dog in the home, each of you shared more microbes with the dog than you did with each other. It's a saying, that says something about people. But this really points out to us, in these kind of analyses, we can dig down into those particular taxa that we identify, and we can see what environments they come from. We can see where a dominant animal bacterium may also be found on the home surfaces. And because we're tracking over time and across different surfaces, we can track them down. I did have a cool video. Because I've transferred it to a PC, the video disappeared, so I've eradicated it. But we don't mind, do we? No. It's cool. But this graphic really helps you to kind of get at the complexity of this interaction that we're trying to deal with. On the inside here, we have all of the samples arrayed by how similar they are compared to the relative abundances of the bacteria and their phylogenetic placement. So if a sample is really microbiologically similar to another sample, then it's very close to that sample on this tree. Okay? And the samples that are very far away from each other on this tree are very different. And what we see if we color these tree branches by the house, we start seeing interesting things like this house over here. This is one house, it's house 4. And if we look here, we can see the surfaces which these samples represent. They're samples from a dog's nose, and a little sample from I think it's a dog's foot, and then some samples from a kitchen counter, I believe. And so what we see here is the microbiome of the dog. I think actually, no, that might have been somebody's hand. Is that a hand sample or a front door knob? There is a hand sample in there somewhere, I know that. It's down there, you can't see it. But so the owner fed his dog daily, and his hand was constantly contaminated with, well, I say contaminated, it was enriched for bacteria that looked like the dog's nose, and also the kitchen countertop was, because he has big dogs, and those dogs leap up on the surface and nuzzle around looking for food. And so you could see that interspertial relationship. When they moved into the new house, we saw that the dog's bacteria colonized that house a hell of a lot faster than any human microbes, unless you had children. Now, if you have children under the age of 5, their microbiome suddenly increases, especially the microbiome from their nose. And I had my own family done, because that's a prerogative I'm allowed to do. And when we see it, my son, my youngest son, who has a severely dribbly problem, massive overproduction of saliva, his oral microbiome was spread everywhere in the new house that we moved into when we shifted very, very quickly. I mean, literally over 1 or 2 days, you saw this oral microbiome on door handles, on the floor, on light switches, and the microbes from there were unique to his oral cavity. None of the rest of us had them. So we were finding that relationship spreading. Now you can track, this is a young couple moved into an apartment in Chicago, a man and a woman. They're about 27 years old, each of them. They have no children. They have a nice new apartment, and they're living the high life. And you can see that both their feet were heavily contaminated or heavily enriched for Staphylococcus. That's quite a common skin-based microorganism. There are pathogens in that family, but mostly this is a non-pathogenic group. And you can see on the bedroom floor and the kitchen floor, after about 5 to 6 days, that floor also became dominated by Staphylococcus. So they were imparting their microbiome into that environment, and it was persistent. Now, this may be, you think it may be cursory, but if you go into a hospital and walk around with microbes in that environment, and then you come back home, how persistent are those microbes on you if you've interacted with someone with a severe pathogen? How long does it stay with you? How quickly do you transfer it into a new environment? And how persistent is it in that new environment? These are all questions which fundamentally we still do not know the answer to. We still lack that epidemiological understanding of the transfer rate and of the occupancy and epidemiological baseline. A lot of this you can find on homemicrobiome.com. This is another big push. Open science is very important to us. All this data is publicly accessible, and we want to make it as available to the public as possible so they can see it. To that end, and that epidemiological study, we pushed forward with the Hospital Microbiome Project, which is a couple of $1 million initiative to investigate an $800 million hospital. We were lucky enough to be able to get into this new hospital before it was open. So we started sampling this hospital at the beginning of this year. There's a MySeq run currently running at Chicago with the first 2,500 samples on it, and I can't show those to you because they're still running. That's not Illumina's fault. I'm not blaming you guys. Maybe. But it's just that won't be off until the beginning of tomorrow, and then we'll have those results. So if you come along to the programs on a Sunday, then maybe you'll see some of those results. But in essence, this study was to look at patients and their rooms daily with 10 rooms over a 365-day period, with some of those rooms during that period sampled hourly. We are looking at, on the patient, the nose, the armpit, and the hand, and in the room, everything. Floor, bud rail, air filter, cold water, glove box, surfaces around the bed. Also, we're collecting bioaerosols from that environment during and after cleaning events over prolonged periods of time, trying to understand how the microbiome changes. And we got an interesting line here because the top floor is cancer ward. It's cancer and tissue transplantation. The patients in there are in there for weeks, sometimes to months. And so they have a very long duration and occupancy. And the patients on this floor are elective surgery. So they're generally in there for like a day, sometimes 2 or 3 days maximum. And that borderline difference, despite the fact that architecturally and from the building science perspective, these two floors and the rooms are absolutely identical. They're on the same water risers, the same HVAC system feeds into both of these sets of rooms. And we analysed both of them over the whole time period, so we could start to understand if you have someone in that room who has a Clostridium difficile infection for 3 days, and then someone else comes into that room and they've just taken a massive dose of antibiotics because they're about to undergo a gastrointestinal surgery to have a benign tumour removed, then we go into that system, we look at it, and we can see what the percent probability is that that patient who's moved into that room will have will gain an infection. How persistent is Clostridium difficile in that environment? And so we've been working with people doing fecal microbiome transplants to actually investigate this in the houses of people that undergo these diseases, and now applying that technology to the hospital. This is 15,000 samples, including staff, and including the nursing stations in which the staff work. 15,000 samples over a 1-year period. It's incredibly intense. The postdoc has moved next to the hospital so that they can be in contact with the hospital daily, 365 days of the year. And we had done some preliminary sampling and analysis, and we see things before anyone moves into this hospital environment, like the cooling tower water looks like if you swab the toilet bowl or the shower head, it looks the same. Those microbiomes are persistent in the water system. But then the water and the water drains from the cold and hot taps and the actual drain themselves, they're actually quite different. They show a bigger relationship to each other. Interestingly, the operating room floor is very similar to tabletops and floors throughout the hospital before anyone's moved into it and is utilising it. If you take people on a tour of the hospital, this comes to how much do you pick up, we see a significant increase in the microbial diversity of their shoes as they walk around that hospital, and the microbiome they pick up from the hospital is persistent. And it persists for about a 6 to 7 hour period before those microbes are overrun by whatever else colonizes the soles of their shoes as they're walking around. We've done this with graduate students from the University of Chicago, making them sample themselves hourly for a 48-hour period as they go through these processes. You see amazing changes. It's quite awkward for them. They had to walk around with kits all day, and wherever they were on the hour, they had to stop, swab the floor around them, swab the soles of their shoes, heel and tip, swab their phones front and back as a backup, and then keep on walking. And they actually got recorded in some of the local newspapers as what the hell are these people doing? But that goes to show you the lengths that the graduate students will go to for science. So we're trying to explore that relationship and see how that happens. And again, you can read up a lot about this on hospitalmicrobiome.com. We publish the proposal, the entire project outline, any publications that will come out of this work, and also some preliminary results and public forum analyses that help us to engage with the public about not just the worry that there may be a pathogen in this environment, but mapping those highways and byways of this ecosystem. What effect does that have upon the entire ecosystem? That brings me on to American Gut. This is our ultimate public outreach. So this is Rob Knight down at Colorado, who you'll see later, and myself at Chicago, and Jeff Leach down in Louisiana. And we've set up a public access forum where we've got thousands of people from America to sign up, give us $99, and we send them a kit. Very similar to 23andMe, if you've ever done that, I have. They send you a kit, you spit into a tube, you send it off, and they array the DNA against about 1 million different SNPs on a microarray and give you the results about your likelihood of getting certain genetic conditions, even if you have wet or dry earwax, things like that. Well, in this, we're looking at the microbiome of oral cavities and of fecal samples. People are even swabbing their dogs now. We get a lot of dogs. There's a dog population out there, much more than the cat population. I don't know why. And they can come onto this website and they log in with their details and they see their sample in comparison to the rest of the samples in the system. Currently, there are about 500 or 600 so processed. We should have about 2,000 or 3,000 processed by the end of May. Those are in the pipeline. We just got to get them through a system and send them to the participants before they're put online here. And we're continuing to fundraise for this, so we continually go through the system, and you can just go and give, and people are donating this to their spouses for Christmas presents and birthday presents. It's very odd. I haven't actually done it myself yet. I probably should. But this enables people to actively engage with their microbiome and identify what's going on. And again, this is all done on the HiSeq and MySeq platforms using the EMP standard protocols. Now, this comes to just a couple of slides to end up with, but the future of the microbiome in medicine. I would like to see a time when we're not just looking at the vital stats of the person, which may intrinsically include microbial processes, but do not explicitly deal with them, and therefore we can't model them or cope with them. I'd like to see a point where you look at the other 90% of your body cells and identify what's going on in there and track it over history. So when you go in for a checkup, you also go in for a checkup for your microbiome, and we see what impact that could have upon processes. In 5 to 10 years, I would stake my reputation on the fact that we'll see the microbiome as an integral part of medicine. And I don't think anyone currently should probably disagree with that right now. To the point where we might get to probiotics, and Jessica Green talks about this a lot, but we've also been a progenitor of this for indoor environments, creating complex microbial swabs that you can just basically wipe down a surface. Instead of sterilizing it, you actually infect it or colonize it with a complex microbiome, which can protect it from all of the pathogens that may interact with that environment. So that's very important to us, trying to understand how we can utilize that. And by teaching people, teaching the public that, as Clotilde likes to say regularly, that dirty things, it's not dirty. Dirty's good. Dirty's clean in many instances, as long as you're not eating your own feces like my dog does. But dirty's good. It helps to increase the microbial diversity of your life. As long as you're doing it in a regular way, increase the biodiversity of your environment. Bring a dog into your house as your children are growing up, and statistically, they have less chance of developing allergenic or immunological responses to stimuli. So less asthma, less allergies. That's a statistical baseline that we can all live onto. If you grow up in a rich biodiversity environment, that's cool. I'm just going to quickly introduce the Earth Microbiome Project. I don't know how much time I've got, probably over now. Fine, 5 minutes. So this is Janet Jansen, Rob Knight, who you'll hear from later. Janet's at Lawrence Berkeley National Laboratory, Rob's here, and I'm at Chicago. We started up this as part of a larger consortium about in 2010. And this kind of splintered off and then evolved into a grassroots project with thousands of collaborators around the world and at least 200 or 300 good collaborators who give us interesting samples. The baseline is that we can't culture all of the organisms. These are some new trees that are coming out of a group in Minnesota. And here we can see samples that have been cultured are in white. So that's a lineage that has been cultured. Everything that's uncultured and has no genomes that are currently accessible is in black. Here's for the bacteria. It's getting a little bit better. A lot of these have been cultured. Most of them haven't been. So we want to look at the uncultured majority. There's a sister project, Microbial Earth, which is going into culture collections and genome sequencing. That's led by Jonathan Eisen and Nikos Kapridis. But our project is trying to get at the unknown unknowns via looking at metagenomics and looking at 16S profiling. We've currently done over 10,000 samples that are publicly accessible at microbio.me/EMP. And if you go to that website, you can, microbiome/EMP, you can see all of that public data. It's all accessible in a public forum. Anyone can download it, anyone can use it as they want, and that's donated from over 134 collaborators from around the world who have visionary thinking and don't mind making their data public. In this instance, these are unknown unknowns. This is an organism which we have no idea. Phylogenetically, it belongs to a brand new phylum, but it's hyperabundant in creeks, in humans, in ocean systems, in freshwater lakes, in rivers around the world, and we even find it at a very, very low abundance in some soils. This organism, 100% nucleotide similarity, is found in both Russian taiga and in tropical forests and in forest soil from America. So you've got this organism which appears to have global distribution. A caveat based only on the 16S ribosomal RNA sequence with 151 base pair fragment. So it may not be the same organism, but it gives us a good phylogenetic basis to track that organism down and find out what it's doing. So we're quickly working to take shotgun metagenomic data and this 16S database that we do have and try and reassemble genomes from those things. I stole this from Jill Banfield, who I was talking with on Tuesday. So we can recreate the metabolic capabilities of those organisms, and through our large database of tens of thousands of environmental samples, put those organisms in an ecological context. Find out what environmental gradients they cover across. Find out if they're predominantly found in high ammonium environments or predominantly found in low pH soils, predominantly found in the oral cavity of people from Malawi. We can do that because we have literally tens of thousands of environmental samples from myriads of different environments, all processed in exactly the same way. And Rob's going to talk about that in a bit later. So the Earth, I could show that, but I'm not going to. The Earth system model, this is our global vision for what we want to do. This is based on taking predictive models of the global climate and then forecasting our ability to predict how the Earth may be changing and what impact that would have upon the microbiome of the planet. So this is predicted values for soil moisture across the globe. We take soil moisture and about 12 other different parameters and use that to predict from the Earth Microbiome Project data the global distribution and a relative abundance of certain organisms. So for example, this is alpha protobacteria. We can do all the other classes or family level assignations based on the EMP data. We can then take that and we can use a new tool we've developed called PyCrust to predict over 2,000 different enzyme functions and their relative abundance. If anyone wants to learn about PyCrust, come and talk to me afterwards. But this is the relative abundance of cellular bicidase in January 2005 across the globe. And then if we know that, we can use some new techniques we developed called predictive relative metabolic turnover, or PRMT, to predict how that community is changing the capacity to consume or generate different metabolites. So this kind of modeling framework allows us to generate rather, some would say, spurious predictions out into the future. So for example, here we can spin this relationship and predict what the microbial community may be, how it may be responding to different climate change scenarios. RCP 4.5 is an everyone board a Prius scenario, a moderate reduction in CO2 emissions. And RCP 8.5 is a business as usual scenario. Everyone's constantly producing the same amount of CO2. And we can now generate predictions of what the microbial portion of, say, the soils or the oceans are doing in terms of their ability to, in this case, consume or generate carbon dioxide. So we're using the EMP microbial data not just to generate a database, a knowledge base, but also to create predictive models that help us to understand how they're responding to different ecosystems, generate hypothetical scenarios whereby we can determine what areas may be carbon sources or carbon sinks in the year 2100. So we're trying to put a practical spin on this, an application. I come from an industrial and governmental background, and so I've always been trained to push in that direction. But I still believe this is fundamentally important. Grassroots academic science can lead up, and if we have the right tools, we can create these kinds of scenarios. Okay, that's it. I covered a fair bit of ground there, but hopefully you get the idea.
Thank you very much. We will have an opportunity for questions at the end of each talk, and I have a question for you, Jack. So in the home microbiome and the American Gut Project, sort of the model of citizen science where you're asking people to contribute samples, how did you raise awareness, or how do you go about raising awareness for these types of citizen science projects, and how do you think that's going to impact your research in the future?
You get very good at talking to the press, and you make very good friends with people who write blogs for National Geographic or for Wired magazine, or people that are actively in a situation where they can reach out to the public. Our ability to communicate with the public is sometimes limited by our own heightened sense of the importance of the nano facets of our research. So we're very interested in working with people who can help us look over the details and come up with a summary which enables us as researchers to transcend that information barrier and say, well, this is the key finding. This is what's interesting to the public. And then, by doing that, we got people that are fundamentally interested. But also, we're riding the crest of a wave here. The microbiome is becoming very rapidly fundamentally interesting. Michael Pollan just wrote an article for us in the New York Times, detailing our work in the Earth Microbiome Project, and that in the American Gut Project, and that's fantastic. Reaching out to the New York Times readership in a front page magazine article, people read that and they go, oh, well, the microbiome could be potentially interesting. Michael had a bit of a tendency to maybe romanticize some of the aspects, such as returning to eating grains and plants. But the idea is still there if you get people excited about it. And we've had so far 7,000 people sign up. And they're paying their own money to help advance a research agenda which potentially should be being paid for by NIH or other organizations, but for which we need to provide this baseline, to provide a large database against which we can ask statistically relevant questions and then move forward with directed experimental design. So the community sees that as an advantage, I guess, while also getting something out of it for themselves.
Other questions?
I bamboozled you. Oh, we got one.
Great presentation.
Thank you.
So you do 16S. You referenced the 16S profiling technique of the 9 hypervariable regions. What's your opinion on how those should be deployed in a study like this?
So when we get metagenomic data, shotgun metagenomic data, we actually revert to something a little bit more sophisticated. So we developed some protocols that we adopted from Jill Banfield's work, looking at multiple ribosomal proteins and multiple housekeeping proteins to try and identify the phylogenetic basis of the organisms in there. But 16S provides you with a quick, very cheap, and very rapid way of getting at this issue. So if you want to process 20,000 samples from myriads of different environments, your first step is to do an amplicon run, right? So an amplicon run gives you deep coverage and enables you to analyze the community. But in terms of the number of different variable regions, we currently focus on V4, purely because nothing's perfect, far from it. In fact, everything has horribly complicated biases. But it's the one that provides us with the greatest breadth of phylogenetic capture across the hundreds of environments we're looking at. When you're looking at soil, air, humans, animals, plants, marine systems, freshwater systems, hydrothermal vents, everything on Earth, and we're even going to the space station in November, so we'll be just slightly outside of the Earth's atmosphere, but still within inside the Earth's gravitational influence, which counts it as the Earth Microbiome Project. Then you have to have something which is broad scale. So we chose V4 for that, and we also standardized the DNA extraction technique, which has its own biases again. So nothing's perfect.
Well, so that was going to be my next question, is that if you're doing tens of thousands of libraries, has that become the bottleneck? Is the labor involved in making libraries?
The bottleneck is still the DNA extraction. We would like to increase the speed of the library production, but the current bottleneck is it's not even the DNA extraction per se, it's actually getting the samples into 96-well plates, getting them individually laid into 96-well plates, and then putting that plate into the robot to do the extraction. Once they're loaded in the plates, we can process 1,000 samples a day if we need to. We have a couple of robots and we have enough sequencing platforms that we could push that forward. You get them extracted in the morning, the PCRs in the afternoon, put them on the MySeq, and then you're going away. Yeah. But getting them into the wells, that's a really tough and laborious process. And the major bottleneck is getting the samples in the first place. Actually going out there into the environment with a proper experimental design and acquiring samples, that takes a hell of a lot of money. That's the most expensive thing now. And it's the real problem. We can't generate samples fast enough to quench our thirst.
Thank you.
Okay, thank you very much.